Today I'm going to walk you through day 2 of the DDG nutrient testing lab provided by the Kansas corn stem. My name is Brent Connor. I'm a lead teacher for the Kansas corn stem program. Today I'm going to walk you through day 2 of the DDG nutrient testing lab provided by Kansas corn stem. The purpose of this lab is to introduce kids into distiller's dried grains, which is a byproduct of distillation process in the process of ethanol, and livestock feed, and we're going to test the nutrients to see the nutrients between a fermented corn versus a non-fermented corn. So in day 1 we showed you how to prepare the mash, which is the fermented corn. One of the other things that you're going to need is the corn slurry. So today we're going to show you how to produce the corn slurry, which is very similar to the mash. You can either do that the day of the lab, but there'll be a little cooling time that you'll need to take into account, or you can go ahead and do that on day 1 as well and cool that overnight with the mash. So same thing as with the mash, we need 300 grams of ground corn. 100 grams of ground corn, and then we're going to add 300 milliliters of distilled water into that. And we're going to give that a nice little stir. Mixed in. And then we're going to put that in our hot water bath, which is just in I just have a 1,000 milliliter beaker here with some water, and we're just going to set that down inside there and give that a nice little hot bath in there. And we're going to kind of stir that intermittently for 15 minutes until we get that kind of a little thick oatmeal consistency. Okay, so it's been a little over 15 minutes. This is our corn slurry, and it has been able to cool down to about room temperature. If it is too thick after it's cooled, you can just put a little more distilled water in there and stir that up to where it's a little bit more liquidy. It doesn't make any difference as far as you don't have to reheat it or anything. All right, so now in order to get our solutions ready, we have to do we got to filter this. We have our beaker, another 1,000 milliliter beaker. We have our little strainer and some cheesecloth. And so you can do whichever one. We have to strain both of them. So I'm going to start with the slurry. And we just kind of pour that in there. You kind of want to be careful that it doesn't get all over the edges because it will run out the side. But you're just going to kind of shake that in, and it'll drain down through the bottom. And you don't necessarily need all of that, just as much as depends on the size of your classes for that. Once it's in the strainer, then we're going to fold our cheesecloth up, and this is where it gets messy. And you can actually have your students do this. The students actually like to do this part. The biggest thing that you have to be careful of is if they squeeze this too hard, the cheesecloth will rip, and all of your dry grains will actually come out, and you want to try to get as much liquid out as you can without getting the actual materials out of there. And so the way I do it is I just kind of squeeze it a little bit, and then I'll start twisting it kind of like a little tourniquet, and that way I'm not squeezing it as hard at the bottom. And however much material that you need, if you have a large class, you'll need a little bit more. If you're just teaching one small class, that's probably plenty of what you'll need for that. If you do run out of liquid and you need more liquid, you can always just rehydrate that with more distilled water and then squeeze that out again for that. So this is what our corn slurry then would look like for that. And then we're going to take this one and we will distill strain our actual mash for this one. And again, you can have your kids do this one. This is the one now that this is fermented. So this actually hopefully has some ethanol production in it through that fermentation process. So you can kind of see a little bit different. We have some bubbles up there. That was the carbon dioxide being released on that. So this one This one does smell a little bit. Some of the kids don't like getting that on their hands. But you can see this is a lot more liquidy because of the ethanol that was produced. So you're actually getting liquid out of there. So we'll just kind of pour that in, and we'll do the same process. Fold this up. And again, you want to get as much liquid out as you can. And so when we talk about we're checking the nutrients of DDG, this is actually what your DDG would look like. So this would be a wet distiller's grain. So this is the byproduct of that. And so the ethanol processing plant will process this. They'll wash it and clean it up, get as much of that ethanol out as they can. And then this will go back into the farmers, and they'll feed that to their cattle. So this is what we're actually testing with the liquid down here, because if the nutrients were in the DDG, they're in the liquid. So it's a little easier to test the liquid than the grains. But this is why this is important to test, because we want to make sure that what the farmers are getting back to them are nutrient-based materials. Okay. So we will get rid of that. Now for the lab itself, I will then take our corn slurry and our corn mash, and put those into smaller materials so that each lab group will get some of those. So that is what is inside these little vials here. So I just put an M for the corn mash, and the corn slurry, yeah, you can put an S on there if you want, but just so they know the difference between the two on there. So to actually start the nutrient testing process, the materials that you'll need your corn mash and your slurry. You will need 6 test tubes that are labeled. We're actually going to do 3 different nutrient tests. We're going to test for glucose, carbohydrates, and protein. So I will, on our test tubes, you'll need to label 2 test tubes, 1 slurry glucose and 1 mash glucose. So you can compare the difference of those two. You'll need 2 test tubes labeled carbohydrate slurry and carbohydrate mash for that test. And you'll need a 3rd set of test tubes with our protein slurry and our protein mash. So you got to make sure that you want to get those in the correct area. The other materials that you'll need are your 3 solutions. So our first solution is our Benedict's solution, and it's the lighter blue solution, and this is what we're going to do for our carbohydrate testing. The Lugol's solution, which is an iodine solution, that is going to be for our carbohydrate, and our puree solution for the protein test. The first test that I like to start with is the glucose test. That's the only one that you have to heat in order to get it activated. So I start with that one. I still have my water bath going here. So I do have my test tube holder for that. You can test them both at the same time if you want, or you can do them individually. So in order to do that one, this is for the carbohydrate test. You will take your mash, and I always give it a little shake just to make sure that everything is good, and of course it's going to spill everywhere this time. And we are going to take 2 milliliters of this solution, and this is the mash, so we've got to make sure it gets in the correct thing there. Get this cleaned up for us. You can see there's the reason why it did that is there's still some fermenting process going on there. So even after you put it in there, you want to check them every once in a while. Yeah, you can vent them a little bit because there's still some carbon dioxide being produced in that fermentation process. And then we're going to take 2 milliliters of our corn slurry, and we're going to put that in our other test tube. And the one other thing you want to make sure for your kids is not only keeping the materials separate, but also your pipettes. So make sure you're not crossing over the pipette so you don't get any cross-contamination there. So for this test, we're using our Benedict solution. So the Benedict solution, we are going to do 2 milliliters of that as well. So 2 mils in our slurry. And I'm going to go ahead and put 2 mils in the mash as well. And you have an option. You can cover this. I use taller test tubes, so I don't have to cover them. If you have a smaller test tube, you may want to put a little parafilm or even a stopper in there that keeps it from splashing. But you want to just give those a little bit of a shake. And then this is the one that you want to have your test tube holders, because this one these go in the water bath. So we're going to set that one in there. And they go in the water bath for 2 minutes. So you want to make sure that your students have someone there to time those. And what we're looking for is any type of color change. So this is when I would introduce our student sheet here. So this is inside the materials that you'll get. So it goes through the procedures of each one. But then on this, on student page 3, here is our chart that we're going to keep. So we have our corn slurry versus our fermented corn mash. And this one is our glucose test, which is here. So any color change that you note on here is what you are going to put down. We're getting pretty close. All right. And we're just going to give those a little shake, and then we're just going to bring those back, and we're going to let those sit for a little while and kind of see where we're at on that. So we'll just let them sit for a few minutes, and we'll check those. As those are sitting, we can go ahead and move over to our next one, which is our carbohydrate test, to test to see if there's some carbohydrates or some starches in there. Again, we have our 2 test tubes that were labeled, and this time, this only calls for 1 milliliter of each of the slurry and the mash instead of 2. So there's my mash. And my slurry. And for this test, we're using our Lugol solution, which is an iodine solution. This, I always make sure, you gotta tell your kids to really be careful. Iodine will stain pretty much anything it touches. You know, depending on your class, if I have some lower level SPED kids, I will go and do this one for them, or have some of my higher level kids come by and drop this in for those students, just so it's a little more safe for them. And for this one, we just need 1 drop of that. So I'll just pull some up, and we just need 1 drop in each one of those is all that you'll need of that solution. And this one you do not heat, so got some on the edge there. And so we just want to shake that around a little bit. And again, we got some color change that we want to make note on our sheet. Definitely some purple in there, some dark purple. So not only do you want to do the color change, but you also want to do any type of darkness. So you want to dark versus light, because the darker it gets, the higher the quantity that could be inside there. We don't necessarily go with quantity on this lab. This is more of a qualitative lab. Was it there or not? So we don't necessarily have to go in, but I just tell them that's a descriptor that they can test, kind of compare with on that one. So I'm going to go ahead and go back and check this one as well, see if there's anything. It's kind of hard to see with the camera, but there's a little bit of a red precipitate down in there that's starting to form, so we'll let that sit a little bit longer, see if we can't get that to come out for us on there. Then our third test is our protein indicator test to see if there's any protein in there. Again, I have my slurry and my mash test tubes, and for this one we are going to add 1 milliliters of our mash and our slurry again. There's our mash. That's why I like to do this lab at least in partners. That way each partner has their own test tube. If you do more than 2, they can get bored pretty easily. So partners of 2 usually works pretty good with this. You're not using very many materials, so it's pretty readily available. And then for this one, we're using our puree, which is the darker bluish purpley solution, and we're going to put 2 milliliters of that in there. Again, making sure we're using a new pipette that has not been used yet. And there's 1, and there's 2. All right. So we're going to just give that a little shake. We're going to let that one sit a few minutes as well. Again, noting any type of color change as that goes through. And so as that's sitting, again, they'll still do some change as we go through. So as that's sitting, I usually will just go through my PowerPoint a little bit, kind of talk about some things. First one is, why is this important? We kind of already told them, the nutrients inside those DDGs, those grains, are important to the diets of livestock. We got to make sure that we have the correct ratios so they're not getting overly neutralized or under nutrient is a big one as well. Okay. And I did forget to go over this. We will do some pre-lab questions. What is fermentation? What role does yeast play in the fermentation process? If this is a standalone lab, that might be a little harder. You're going to have to do a little bit more pre-work on that. If this is the lab that you're going to use to follow up distillation, they should know some of those questions because they've already used that to get to that distillation process. So that's why I usually prefer to do this lab after the distillation on that. And again, we go through on the PowerPoint every test just like we did. As we go through there. So then our starch test, our protein indicator test, and then our data table. So at the end, after they have completed their list, we just, again, have them do a final check, see what we have. So a little bit different color there, kind of turned to grayish, almost a little brown in the mash. So we'll kind of compare the difference of the color changes there. Those two haven't changed much. So we got a little light purple and a real dark purple there. And then we have that. Not a whole lot of red precipitate in there, but just enough in there that they should note that one as well. And then our lab analysis then, from our pre-lab questions, were your predictions correct? Was fermentation successful? Is it vital to the health of the cattle? And then the results of what happened during that fermentation process. So they should note, that there was some, the sugars were more readily available, and they kind of get eaten a little bit by the yeast, but that becomes a nutrient then at the end for the cows and the cattle. Reflection and conclusion, very important. Our livestock are needing of the protein, so the protein does show up in the mash. for that reason. We kind of go through macromolecules again, depending on the level of your kids. Sometimes that's as far as I go. If they're upper-ended kids, higher level kids, then I'll go into a little bit and we'll talk about the micromolecules and all that. And then you can even take it farther, do an enrichment type thing. We'll kind of do a little research between the difference between a wet distiller's grain and a dry distiller's grain as far as uses, effectiveness for feeds, longevity, that kind of stuff. And they'll just write a 2-paragraph summary versus the cost of feed, the availability of that feed, and the nutrient supply of that feed. Then you can even go even farther if you want and talk about the different careers that are associated with that. So ag inspectors, specialists, chemists, food specialists, all those type of entities are very high in this nutrient testing. So that will conclude the DDG Nutrient Testing Lab. If you would like to see more labs, you can visit kansascornstem.com.